need to agree the recording starts.
And there was one of the angle so that people have these face of the use green.
Yeah.
Then yeah. How was yours?
Yes.
What was the people that say that you could see they should be
Okay, welcome everyone. Zoom audience, let us know if you can't hear. We are monitoring the chat. It's a great pleasure to introduce George Meliaros. George is the Prince Philip Professor of Technology at the University of Cambridge. He has a PhD from the University of Groningen, and he taught at Cornell and he called the mines before joining Cambridge. And we look forward to his talk. If you have questions for online audience, please put them in the chat and we'll read them at the end. Thank you, George.
Thank you very much. I appreciate the invitation. I see a lot of complementarity between the groups, and I look forward to a lot of collaboration. So today I'll talk about implantables, which is the main activity of the group. And it's also a very data-heavy endeavor. So I'll talk about hardware, but we do need help and collaboration here as well. In addition to the wearables. So technology for bioelectronic medicine, I'll explain what bioelectronic medicine is and why it's in need of technology. Let's start with a broader picture. What is medicine? And it goes as follows. As we go through our journey from birth to death, the length of that journey is determined mostly by the genetic material we inherit from our parents. But there are factors that can accelerate that journey. For example, for diets, pathogens, very much a la mode with the pandemic, accidents can shorten our lifespan. In this context, medicine is the set of tools and know-how that we as humankind have developed to cheat death and turn back the clock and get more quality of life. How does that work? What are the modes of intervention? The most traditional one is pharmaceuticals. You feel sick, you go to the doctor, and they will prescribe some drugs, typically used for prevention, symptoms control, or for curative outcomes. A couple of examples here: vaccines can help you getting sick. Administration of dopamine in people who have Parkinson's disease will control the symptoms, will not cure the underlying pathology. And antibiotics will kill the bacterial agent that causes disease and yield a curative outcome. In the same vein, surgery, radiotherapy, where you go into the body, you cut and connect and burn, can be used also for a variety of outcomes. Removal of breast in people who are at risk of developing breast cancer is used for prevention. The installation of a tube between the brain and the abdominal cavity can drain excess cerebrospinal fluid in hydrocephalus. And the resection of a tumor before it has had the chance to metastasize gives a curative outcome. Now, this is traditional medicine, the way it's being practiced. In these modalities, there are two new ones that were added in recent years. One is the regenerative medicine, where you go into the body and add tissues. So these are tissues that come from humans. You get cells, you change them into a state that you want them to be, and then you go and put them into the body. And this has been used to treat, for example, leukemia with a curative outcome. In the same vein, you can go into the body and add an electronic device, which through electrical stimulation can have a variety of outcomes. So that's what I'm going to talk about. It's called bioelectronic medicine. And one example is a device called an implantable cardioverter defibrillator. Don't know how many of you are in football. This was from a while back, 2 summers back, when 3 summers back now, when the Euro was going on. One of the Danish team footballers collapsed in the field because he suffered cardiac arrest. The heart stopped beating and he fell on the ground. And he was taken to the hospital and fitted with an implantable cardioverter defibrillator. So this is a device about this big that gets placed on the skin under the chest here, under the skin here on the chest, with electrodes that contact the heart from the inside and monitor the beating of the heart. And if the heart stops, then it's going to give it a 750 volts boost and restart it. So that's called defibrillation. And Ericsson now plays for the Premier League. So he's back in full performance. Having given that example, which is from the area of prevention, most bioelectronic medicine interventions today are for symptoms control. This is one example. This is a patient who suffers from a movement disorder called tremor, which, as the name implies, it's associated with uncontrolled movement of extremities that makes life difficult. And through electrical stimulation, the symptoms ameliorate. It's not perfect, it's not a cure, it's a treatment, but it works reasonably well. So here the intervention is delivered by electrodes that are placed in a particular structure of the brain called the subthalamic nucleus. And this has been used to treat movement disorders such as dystonia, Parkinson's disease, tremor, has been used to reduce seizure frequency and intensity in epilepsy, is used for neuropsychiatric disorders, severe forms of OCD and depression. It's being trialed in obesity and a variety of other indications. This goes way back in time. In classical times, people were using electric fish to deliver shock to help people with migraines. It's not clear what the efficacy of the treatment was. There you have it. It was Galvani in the 18th century who did the first scientific experiments with a dead frog, applied electricity, got the legs to move, This gave rise to lots of speculation about the nervous system, the muscular system, but also gave rise to medical devices. 200 years later, there were pacemakers. At that time, this is 57, the year before these devices became fully implanted. At that time, the patient had to carry with them this card of batteries and electronics. So in 58 was the first fully implanted pacemaker at the Karolinska Hospital in Sweden. And that starts the clock for modern bioelectronic medicine. So here, the location of the intervention is the heart. You're pacing the heart to make it beat at a regular rhythm. And then a decade later, stimulation of the inner ear in the form of a cochlear implant became available to help people with profound deafness. Then spinal cord stimulation for treating chronic pain in the 70s. Then a couple of other peripheral nerve devices. The brain came to the forefront in 2000 with deep brain stimulation for neurological and neuropsychiatric disorders, and then it exploded. So many, many devices. Not a comprehensive chart. The latest and greatest is peripheral nerve stimulation to expand the scope beyond neurological and into autoimmune diseases. For example, Crohn's disease, type 1 diabetes, rheumatoid arthritis are all believed to be treatable with stimulation. And it has rose to a level of prominence where big companies such as Google's parent company and GlaxoSmithKline launched ventures in this area. This is Galvani Bioelectronics, about half an hour from here in Stevenage, envisaging a future where you treat disease through electrical stimulation. And then, of course, the founder of Tesla, the founder of Facebook, they all got into this game. Lots of hype. If you look into movies, it's always a bad man with bioelectronically controlled tentacles that is portrayed as the picture of the field. This is the clinical reality. This is a boy fitted with a cochlear implant. This is a device that has two parts, an external bit that picks up the sound with a microphone, processes it, and then radios power and data with this antenna to an implant in the inner ear that stimulates hair cells and gives rise to the sensation of hearing. And look at the marvel in the face of this boy as he hears for the first time. So we're talking about life-changing technologies. And any medical technology carries ethical issues. Here, because we're often acting on the brain, which is the seat of consciousness, the organ that gives a sense of self, these ethical issues are compounded. So on the left, you see a picture from an old movie, which is very funny, that portrays the quintessential mad scientist conducting experiments in an ivory tower, trying to galvanize, as it was called, or reanimate with electricity a dead corpse. And the villagers are outside the tower with pitchforks trying to get him. That type of stereotype portrays the picture of a scientist who is disconnected from society. And this is not how medical technologies develop. So we arrived at having devices such as this one, the cardioverter defibrillator, through constant and very early dialogue with all the stakeholders in the game, patients, their advocates, clinicians, industry, ethicists, regulators. So if there is one thing to take away from my talk today is that this is a game of many stakeholders, and you should be engaging with all of them from the very early stages of technology development. So with that as a very broad introduction, let's start now zooming into some of the challenges. This is a cartoon displaying different implants in the clinic today. The location of the intervention would be at the end of this wire where the electrodes would be. And it shows the indications that are being addressed with this technology. So anything from incontinence to chronic pain to disorders of the heart, of the brain, are addressable with this. By and large, they look like this. They have a little box. This is called the IPG, implantable pulse generator. It's about 2/3 battery inside the box and 1/3 electronics. And then on the outside, there is a big connector that connects to extension wires, then another level of connectivity, and then the electrode arrays themselves. So here there are two, because it's a bilateral implantation, one in each hemisphere, 4 electrodes per array located deep in the brain. So that's the electrode array here. These are the extension wires with a lot of slack to allow for movement, connection, and then this goes under the skin of the neck to the chest where the IPG is placed. The implantation of this is permanent. You don't want to move it. There are people with implantable electrodes for over 20 years today, and this doesn't move. This has to come out every couple of years to replace batteries, typically 5 to 10 years to replace batteries. And you disconnect from here, you take it out, put a new one in, connect it, and that's it. Now, the front for research and for development is where electronic materials come in contact with tissue. That is the single most challenging bit of the device. And in all of these devices, it looks pretty much the same. So you have electrodes that are quite macroscopic, millimeter size. They're made out of platinum with a little bit of iridium to adjust mechanical properties. And they're encapsulated in an insulating matrix, typically a polyurethane or a silicone. So this is a cochlear implant. That's why it looks, the shape it has is to get into the cochlea, which is helical in shape. These are different electrodes for the spinal cord, but basically their characteristics are very similar. And surprisingly, they're made by hand. So in the year 2024, if you go to Medtronic, Boston Scientific, all the big companies, that's how their fab looks like. It's people working through microscopes with tweezers, putting things together by hand. It's incredible, isn't it? This is like watchmaker technology of the old, like mechanical watchmaking. While microelectronics is being fabricated using highly automated process, this is TSMC in Taiwan, you see very little space that's occupied by people. It's mostly empty of people running 24 hours a day, using processes such as film deposition, photolithography, etching, and producing objects of incredible precision with every generation lower cost per unit of the item. So this technology now, microelectronics, is slowly entering the field of bioelectronic medicine. There are pictures with blood and guts, so I have warnings if you're sensitive. And it's intersecting another technology, which is that of large, flexible and stretchable substrates. What do I mean by that? These are two examples of that work. This is from the US, the group of John Rogers, who developed large area substrates that are so thin that you can put electronics on it, but if you pick it up to put it on the brain of the patient, it will collapse under its own weight. So they had to develop technology by using sacrificial silk fields to allow you to pick it up and transfer it and then dissolve the silk away. And that leaves you with a tattoo-like structure that follows perfectly the curvature of the brain. So it's like tattoos for the brain, the surface of the brain, giving you excellent mechanical contact with zero mechanical loading. This is from Stephanie Lacour's group in Switzerland. She used elastomeric substrates that are stretchable. And then you can develop electrodes on them, and they follow the motion of the body. So if you put them on the spinal cord, as you go around your daily activity, the spinal cord will expand by up to 10, maybe more, 15%. And these arrays can follow that motion. So combining this flexible and stretchable with microfabrication, you can make electrode arrays that are very thin, flexible polymers. And in this case, with enough mechanical strength to handle their own weight, with making very small electrodes to get very high resolution recordings from the surface of the brain. These are to be placed on the surface. And then this is what Elon Musk's company does, Neuralink. They use a similar technology to make devices that get pushed into the brain. So these look linear arrays with small electrodes shown here. Again, the size is of the order of tens of microns, commensurate with the size of a neuron, the soma of a neuron. So they can record single neuron activity. And those are pushed into the brain, shown here, to penetrate into the brain using a sewing machine type of implement that picks up the implant through a suture loop here and just pushes it into the brain, and then the needle comes out. And this is a technology that got approved by the FDA last year, and now they're in first-in-human trials to help people with paralysis. The idea is to read signals from the brain and then transfer them to a computer, so they establish an interface that bypasses an injured spinal cord. Now, a bit of science here. In addition to using lithography, you need to use a little bit of material science to make such small electrodes. Why is that? If you want to couple between electronics and biology electrically, the challenge is to connect ionic signals in the brain with electronic signals in metal electrodes. So the idea is the following. Our brain is filled with salt water. When neurons fire action potentials, that ions in that electrolyte get set to motion. They undergo an oscillation, which is the spatiotemporal summation of all activities of neurons in their neighborhood. So if you present a metal electrode in the proximity of the brain, inside the brain, what will happen is, as ions move, electrons inside the metal will also move to mirror that motion. And what you detect is called the displacement current. It's a current that oscillates, and you get information about neuronal activity. In the geometry that I just described, that coupling is along two dimensions. You have ions on the side of the brain, electronic charges on the side of the metal, and they only interact across the surface. There are materials, we're not going to talk about details, where ions penetrate into the electrode and couple in the whole volume. So now the whole volume of the electrode becomes a place of interaction between ionic and electronic charges. And this helps you get a better contact. Never mind the details. Consider these materials as spongy conductors where ions from the electrolyte can penetrate and couple. And that is characterized by capacitance that scales with the volume. These are all details. The end result is, let me skip this, is this. You can go today from very coarse to very fine tools for mapping the brain. So this is the incumbent technology that you use in the clinic. for mapping the brain. This is an operation that takes place before any brain surgery is performed. So they will do, for example, a craniotomy. They will expose the dura, shown here. They will then do a durectomy, as shown here. They will flip over the dura. That exposes the surface of the brain, and then they will put an electrode array, again, made by hand with platinum electrodes that are millimeter in size. And they do that in order to record and stimulate the brain and map it before they resect a bit of it out. So this gives you very coarse type of mapping, much poorer than a surgeon can resect. So by using this new technology, you can land hundreds of electrodes in that area. The electrodes are 3 orders of magnitude smaller and can pick up single neuron activity. So a big advancement not only in helping surgeons resect, but better, but also giving you more information about how the brain works. And you can use the same technology to also stimulate and elicit more articulate motion. So these are microfabricated electrodes that go around nerves, and they're called cuff electrodes that wrap around nerves. And here are the nerves of the brachial plexus, which are the nerves that control hand motion. So these are placed on the front foot of a rat, and by stimulating at each electrode, you can get very articulate motion of the arm, much more so than possible with any other larger technology. So these are different motions, for example, wrist flexion or extension or what have you, each one elicited by different electrodes, and it's a very interesting era when we can interface with the nervous system at very high precision. So I'll show you now 2 examples of work, one coming from the intersection of bioelectronics with soft robotics. Soft robotics is illustrated here. This is an example from George Whiteside's group at Harvard, where they make objects out of elastomeric materials. These are silicon rubbers. And by pumping in fluids, they can get these objects to change shape. As a result of the change of shape, this object walks. But that's not the interesting thing. The interesting thing is that it changes shape. So the shape of the object now can be programmed dynamically. And that's something you can use in bioelectronics. For example, spinal cord stimulation is an area where this comes in handy. So spinal cord stimulation, again, is a very established neuromodulation technique. It started in the 70s to help people with chronic pain. And there are 2 electrode designs. One is these linear, so-called percutaneous arrays that are placed on the spinal cord through the space between vertebrae. A clinician will kind of feel your back, figure out where that gap is, jab a needle, and then put this array up the spinal cord through that needle. That's a procedure very similar to having an epidural injection or to having a spinal tap. You access the intrathecal space. So it's done typically by an anesthetist. They will put the needle in, put the implant out comes the needle. And a couple of hours later, off the patient goes back home. Unfortunately, these electrode arrays can move when the patient, for example, bends over to tie their shoelaces or does some acrobatic maneuver. It comes out of alignment, and they need to go back in for a vision surgery. An alternative is this percutaneous array. Once this is placed, it covers a larger part of the spinal cord, delivers treatment over a larger area, and also doesn't move, it stays there. Unfortunately, to put those in, you need the laminectomy, you need a neurosurgeon to cut part of the vertebra out, place the array, and then figure out how to patch that hole. And that's typically associated with a 3-day stay at the hospital. So it's much more expensive and much more invasive to the surgically to the patient. So if you combine now this thin, flexible bioelectronics with pneumatic actuation, you can make devices that look like paddle electrodes, then roll them up to become percutaneous array, put them into the body when they're rolled up, and then expand them once they're in. So the concept is shown here. You do a lumbar puncture, you find the space between vertebrae, put the implant in through there, and when it's positioned, then you can expand it pneumatically to cover a large area of the spinal cord and become a paddle. So this we validated in animals and in human cadavers. This is the lumbar puncture. The device is coming in, and then it's going to get pushed up under a fluoroscopy machine that lets you, through X-ray illumination, find the device. And then once it's in, you can expand it by pressurizing it mildly with a syringe, a saline solution. So this is shown here. That's the needle. This is the device coming out and pushed up. It has 2 X-ray markers, one on the top, one on the bottom part of the device, and they will separate as the device is unrolled, which you see here. And then you can go in and do a laminectomy. So this is where the device was injected. It ended up being placed here, so you can open up and you see the device hugs the spinal cord very nicely. This one here, that one there are the 2 X-ray markers that separate it. So this is now on the path to commercialization. We hope to start first in human within, fingers crossed, a year, if all goes well. But there are other modes of actuation as well, including electrical actuation. This is shown here. These used to be called artificial muscles. It was a field that boomed in the 80s and 90s, and then it dropped off. But now it's slowly coming back. And the idea is to combine a very thin metal film with a conducting polymer. This is a material that can uptake ions from an electrolyte if you apply a suitable potential and swell. And as it swells, it can change the shape of this bilayer structure. So if you engineer it well, it can do really interesting acrobatics. So this is a bilayer that opens and closes under the actuation by a small voltage. So what happens here is you apply a voltage, ions would come into the film, you see a current that flows, and then you would change the angle at which the film is curved. By engineering the shape of that conducting polymer, you can get the implant to do different motions. For example, here is a helicoidal type of rolling motion that was engineered by having fingers of conducting polymer at a slight angle with the axis of the implant. And by controlling the angle, you can control the pitch and all that. So this is a device that you can present to a nerve, as shown here, and then get it to wrap around the nerve very gently. Grab hold of it very, very gently. And you can also integrate electrodes that will monitor that nerve. This is to satisfy a clinical need in intraoperative recordings. So when a surgeon operates in an area that has nerves, they have to monitor the nerves. And they do this with an electrode that looks a little bit like a chopstick, the same size. They need to land it on the nerves and hold it there. And the electrode itself represents a danger to the nerve because it's one wrong movement and it can damage the nerve. So this device can go in very gently and grab hold of it. As it grabs hold of it, you see no neural activity elicited, so it's very gentle to the nerve. But then if you can press the paw of the animal, this is on the sciatic nerve, then you can catch the nerve activity as it travels towards the brain. So it's a device for intraoperative monitoring. So that's the interface with soft robotics. Now I want to talk about something a bit more sci-fi, which is the interface with regenerative medicine. So in regenerative medicine, what you do is you take cells from a human, for example, skin cells, fibroblasts, and you bring them to a stem cell state. For example, you can convert them into induced pluripotent stem cells and store them as such. And then when needed, you can forward differentiate them into different somatic cell types. For example, make them into muscle or neurons or glia cells, and then bring them back to the human to undo damage that was caused by disease or injury. And as I mentioned earlier, this gives you curative outcomes. It's something that bioelectronics cannot do today. So what if you were to combine these two modalities and go into the body by bringing in both cells and electronics? So should we be thinking about regenerative bioelectronics? And the idea is the following: you bring into the body an implant that has both electrodes and tissue. And that you make outside the body, you characterize it, and then you bring it in, hoping that the regenerative component, the tissue, will camouflage your electronics and make them more biocompatible, will repair the host tissue where there is damage, and will enhance the communication between the repaired tissue and the implant through a process of reinnervation. So when you bring them together, the host tissue can project and integrate with the implant tissue, and the implanted tissue can also project, if possible, neurites and connect with the host tissue, leading to better communication. And at the same time, the bioelectronic component, the electrodes, will monitor this process of repair, will even guide it through electrical stimulation, and then you end up controlling the repaired tissue. So this is an interesting concept. One question is where do the cells come from? And these are today commercial. So there are companies such as Mark Cotter's, he's here in Cambridge, Big Bio, which is making big waves in this field. They have developed a technology to take these human pluripotent stem cells and forward differentiate them very easily into different somatic cell types. So what you're seeing here on the video is the differentiation of these pluripotent stem cells that start as round cells into nice striated muscle. So you'll see that soon enough, you'll end up having beautiful muscle tissue developed from these cells. So this is a commercial line. You can buy the cells and the reagents, and then in a petri dish, you can make your own muscle. And to prove this concept of regenerative bioelectronics, we use those cells in conjunction with a peripheral nerve injury model. So the clinical context here is the following. If you have a nerve injury which is over 1 centimeter, then the neurosurgeons cannot take the two bits and put them together. It's just too long of a distance. So you end up with a functional deficit. For example, you cannot move your hand. In order to undo this deficit, you might think that I can record from the proximal stump, the bit before the injury. And if I record from that stump, then I can get motor commands that are issued from the brain. So every time the brain says, move a finger, I can pick that command from the nerve and then stimulate behind the injury and get the arm to move. Recording from that nerve stump is very difficult. So we thought that regenerative bioelectronics might be the way to do that. And it is difficult because the nerve will promptly die off and you end up having very little signal coming out at the stem. But the hypothesis was that if we present muscle cells to a severed nerve, then the severed nerve might re-innervate these muscle cells and maintain its viability. So in this sense, the muscle will help the nerve heal and stay alive. And then every time the brain sends a command for firing, that command will come through the nerve and then stimulate these muscle cells. And then we can record this activity by underlying electrodes. So the experimental timeline involves making a flexible device, seeding the cells on top of that, then differentiating ex vivo. That takes a couple of days, testing the connection to make sure it works well, and then transplanting it in an animal model, peripheral nerve injury, presenting the array there. And this is the data that shows repair. So here, what you see is recordings from different electrodes that are placed on the nerve. So the nerve is severed, you present the array, you stimulate the nerve here, and then you measure activity here, different electrodes. These are data from electrodes without muscle that are placed on the nerve. And what you see is just a stimulation artifact and no further signal. And the reason for this is that this part of the nerve now is dead because after injury, the actions have died off. So you can stimulate all you want here, but there is no conduction of the signal. However, if you present the array with muscle on the nerve, and then of course you have to wait a couple of weeks, then you measure this signal, which is the signature of conduction in a nerve conduit. So it's similar to what you would get in a naive nerve that hasn't been transected. So that shows you that the concept of presenting muscle to that nerve conduit helps preserve its health. So it does the tissue repair function that we hypothesized. And then these are the recordings of spontaneous activity. So recordings from one electrode shown here, week 1 to week 4. See, week 1 and week 2, very little signal. But then from week 3, you already start seeing nice muscle activity, which becomes very well defined at week 4. And you can record these command signals very nicely. You can also correlate them with behavior, and they correlate very nicely, but that's secondary. The important thing is you can show that this regenerative bioelectronics concept works, where you can now envision implants that carry both tissue and electronics to do repair and achieve a curative outcome. So that's the main story I wanted to tell you today. And it paints a very optimistic picture about the future. What are the challenges? There are some formidable ones, including stability. As these electrodes are very thin, a small loss of mass can devastate your device. So to learn how this works, we set up ISO-inspired accelerating testing where we take implants, we put them in media that are spiked with peroxide to simulate the reactive environment that is caused by implantation. We heat the media up to 70 degrees. That gives you approximately 10x acceleration in terms of time. Sorry, and we also move the stage to simulate micro motions. And then put different, excuse me, types of implants and run them until they die. So these are 4 different types of implants. Day 0, 70, and 140. And we do failure mode analysis, trying to understand how they fail. So here you see the electrode has first delaminated, detached, and then delaminated. And then the connection is also corroded. And we can measure the amount of material that is transferred from the electrode and all that, and try to understand failure modes. And through these efforts, we went from implants that fail within a year to implants that fail with an extrapolated time of 6 years, to implants that are very stable. These are still going on. I think this has over 10 years extrapolated lifetime. Another challenge is X-ray visibility. As you make this device very small, you put them inside the body and then you cannot see them anymore. They don't have enough material to scatter X-rays. And we ran into trouble with that when we put the first implants in. We couldn't see them. It took us a year to develop X-ray markers that are compatible with microfabrication that we can now integrate to the implants to be able to see them with fluoroscopy. Then there are issues such as implantability, expandability, as they're very thin, they're hard to handle. How do you put them into the body? How do you remove them if they need to be removed without breaking? There is need for specialty circuits that would accommodate the many contacts that you can have in these implants. Electronic packaging, how do you connect to something which is very floppy and flexible, and many, many other issues. But I don't want to end on a challenging kind of theme. This is very promising because these new technologies enable us to make implants with a host of properties that lead to significant clinical benefits for bioelectronic medicine. You can make them minimally invasive, you can make them high precision and all that. And there are challenges to be addressed, but they're all addressable. It just needs time. So with that, let me thank the group. The group is co-led by a neurosurgeon and a neuroscientist, and has expertise in pretty much everything from physics to neurosurgery. People who contributed to today's talk are highlighted. Collaborators in Cambridge and outside, funding agencies, and thank you very much for your attention. And let me put in a plug. This we're organizing here. Here, when I say here, in this building. Thank you very much for helping us organize this. This is the conference that focuses on young scientists. It's 1 to 3 July. You can come in. We'll close our eyes. If you don't have a badge, you can sneak in from the back door and listen to the talks. If you want to participate in the dinners and all that, you would need to register. Right?
Thank you.
Thank you.
Okay, so I'll come closer so that they can hear me. If there are any questions online, put them in the chat. If there are any questions in the room, raise your hand. One question there? Yes.
I have a question. Thank you so much for the talk. I have a question about the BMI interfaces. This is the research area that's been running for more than 30, 40 years, I believe, right? And I'm not following the literature anymore. It's when I did 20 years ago.
Yeah.
One important aspect of it is that to have something meaningful to BMI, you need to have an extremely large area of electrodes. All the examples you've shown also today are area of 256 electrodes which are positioned on a very specific part of the brain. So my question is about scalability of this approach, which will expect and require whole area of the skull not covered by the electrodes. So how you does that?
Yes. No, that's a very good question. So for BMI, what is used clinically today is an implant which is 3 by 3 millimeters square with about 100, 200 contacts that penetrate into a small area of the motor cortex. So the motor cortex runs, it's like a slab that runs around the head in this direction. And it is well mapped, so you know which is the area that controls the arm, for example, the hand and so on, and they target that area, which is again millimeters in size. The next level up is to provide some feedback to the brain. That would be the somatosensory cortex, which sits in front. And that again, they would do with a similar implant. So it's only small areas of implantation are enough to give you enough data for movement of an arm and to give sensory feedback as if it would come from an arm. You don't need to do a full cortical prosthesis. Those are used mostly to understand how the brain works. So in the clinic, typically, you don't need super large areas, you don't need super high density. For neuroscience, there is an arms race, the equivalent of Moore's law. How many neurons can you interface with? And now it's running into millions or whatever. Now there are 80 billion neurons in the brain, so it's a very small number being probed. And I think that's not gonna yield much fruit. Going for more and more and more recording will not dramatically increase our ability to understand how the brain works. I think what is more important to do is to target different areas and explore connections and to look beyond electrophysiology to combine with metabolic activity, to combine with what do the glia do, to understand a bit broader context what happens.
But for this, you need a lot of electrodes, like anyway, right? If you talk about correlation between different parts of the brain, right?
Yeah. Well, the way it's being practiced today is a cortical array that in the clinic would be a couple of centimeters, would cover an area of centimeter by a couple of centimeters, and then a depth probe that would have something like 8 electrodes in the thalamus or so to look at, for example, thalamocortical communication. Implants in the gut with a brain connection also are a very hot thing. And a couple of other parts of the body that get co-implanted with the brain to establish connectivity. That gets a lot of traction today. There are still people who make probes with many connectors and electrodes and try to put as many as possible. But I think this is clinically not particularly relevant. And for neuroscience, it's kind of saturating you won't get much more impact from this.
I have one quick question, and then I'm just going to get a short one. How about the deep, like super deep electrode? Because the electrodes are like on a shallow surface of the brain, right?
So the targets, again, for the clinic, there is a bit of disconnect between neuroscience and clinic. For the brain, the deepest you get in is the subthalamic nucleus, which is quite deep in the brain. And that has been achieved. There are people with electrodes in that part of the brain for 20 years now. In the neuroscience community, they try different parts, go even deeper into the brain, close to the brainstem, to go to the cerebellum, which is not very well explored, in order to understand what is the function of these parts and how they connect to the cortex. Down the road, this might lead to a clinical use, who knows? But it's still very exploratory. And there is good enough electrotechnology that allows you to do it, not with very high reproducibility. So you can do that in animals, you can access different, very deep structures. There are challenges, but not insurmountable. And currently it's very academic research, so not particularly high yield. Thank you.
I have a question.
Yeah.
As a computer scientist, I kept asking myself at which point this could start to converge. And I'm thinking in terms of reconfigurability of your devices, reprogrammability of the devices, communication with the outside.
Yeah.
Can you give us a little view? I think some work from MIT where they had this ingested, it's not implantable, but it's ingested and was communicating with some wireless level. And obviously there are all the batteries issue, but I wonder, what's your view?
So there is definitely that push going from a system that just stimulates to a system that stimulates intelligently. For example, in deep brain stimulation, you can put a ring with an accelerometer, measure the frequency of the tremor, and then feed back to have kind of closed loop stimulation. It works more effectively. Or do what is called chronotherapy, where you deliver different stimulation protocol at different times during the day.
So for the first one, is there communication between the two devices?
Yeah. There is. And you can go even further where you have a companion wearable that senses different parameters and then communicates with a bunch of implantables in a coordinated fashion. That doesn't exist in the clinic, but there are lots of literature on this. So it goes from a stupid system that just stimulates to a system that communicates with something outside to stimulate, to get information and adjust stimulation, to systems that can not only receive information, but also convey information to do a bit of self-diagnosis of the system itself, but also of the biological environment. So in addition to being able to transfer power, you need to transfer data back and forth. Typically done with electromagnetic induction. All clinical devices work with that principle. Magnetoelectrics is receiving a lot of attention today, and ultrasound and even light as a way to couple with an implant inside the body. There's no one solution fits all. Electromagnetic is the predominant one.
Okay, question with it.
Why is stuff still being made by hands, particularly hard to machine?
Yeah, no, it's extreme reticence of the industry to anything that's new, because if it fails, and it's actually not just the implant, it's the companies that supply materials that will not supply to implant manufacturers. For example, as an implant manufacturer, it's very difficult to get hold of resist to do photolithography because it's not, they don't want to risk the implant failing and then the lawsuit being passed to them. There are a lot of examples where companies supplying chemicals were blamed for devices that failed into the body. The lawsuits are incredible in terms of money being asked for damages, enough to bring down the likes of DuPont and 3M and all that. So DuPont had almost lost its business because of some breast implants that leaked.
Okay, thank you very much for coming up to the hour.
Thank you. Thank you.
Thank you for our audience. We'll see you in 2 weeks. And thank you, physical audience.